Hi everyone, in this video we will be talking about the research article that focuses on the depletion of caveolin-1 in type 2 diabetes and Alzheimer's and possible implications of this study. So before we begin, a little bit about me. My name is Sonia and I'm a sophomore at Virginia Tech studying cognitive and behavioral neuroscience. I've always been interested in the brain and particularly what establishes Alzheimer's disease. I chose this particular article because I'm interested in the neural connection between type 2 diabetes and Alzheimer's disease. Alzheimer's disease is a subset of dementia and is a disease that destroys memory and impairs cognitive function. This disease is characterized by an accumulation of beta amyloid peptides. This peptide is created by an amyloid precursor protein. The accumulation of beta amyloid peptides in the brain can lead to senile plaques, which is a trait of Alzheimer's disease. The interesting thing about type 2 diabetes is that it is a big risk factor for the development of late-onset Alzheimer's disease. Not much is known about why exactly it is a risk factor, but this experiment tracks the regulation of a protein called caveolin-1 in brains of T2DM mice and shows how CAV1 regulates APP metabolism. Caveolin-1 is a protein which is important for cell functioning and signaling. There are many studies going on which involve caveolin-1, including cancer research, because it is shown to be a tumor suppressor, especially for men with prostate cancer, as they have a big decrease in the amount of CAV1 that they possess in their body. CAV1 also has other important properties such as vesicle formation and cognitive function. Caveolin-2 is another important component of the CAV family, which can also lead to changes in tissue damage and inflammation. Both of the caveolin proteins are formed by caveoli, which are basically invaginations of the cell membrane layer. This experiment tests the levels of CAV1 in T2DM patients and in DB/DB mice, and if the restoration of CAV1 in diabetic mice can induce cognitive enhancement. DB/DB mice generally refer to mice that are diabetic and express an insulin deficiency, so it is an important model for studying diabetic patients. This experiment goes a step further and also shows that a decrease in CAV1 proteins actually correlates with the amount of amyloid precursor proteins, APP, in an inverse relationship. It is known that the buildup of beta amyloid peptide as well as a protein called tau are precursors of APP, which is a crucial component of Alzheimer's disease. Additionally, this experiment tests the levels of base1 hyperphosphorylated tau species and impairments of the novel object recognition tasks, but we will not be focusing on those particular tests in this video. The materials used to test this experiment are shown on this slide. There are 6 methods to test the hypothesis in this experiment. The novel object recognition test is often used to test cognitive stimulation on mice and how quickly and effectively they respond to the presence of a new or novel object in their familiar arena. So when testing this, mice were placed in a familiar arena of over 4 different days and were allowed to explore 2 objects, one familiar and one new. The second test that was performed was a stereotaxic injection of caveolin-1. After approximately 3 weeks after the injection, the mice were then asked to perform the novel object recognition test again. To verify that everything went well, coronal cross-sections were also taken and stained. Mouse ELISA assays were also performed with mouse brain tissue. ELISA stands for Enzyme-Linked Immunoabsorbent Assay. These were performed using amyloid beta peptides. Sucrose density fractionation refers to the separation of particles through a chemical gradient. After performing the fractionation, results were calculated using a buoyant fraction percentage and an unpaired t-test. The 5th part of this experiment was using human embryonic kidney cell culture. This was tested using the amyloid precursor proteins, which, as we learned before, are a key component of the early onset Alzheimer's disease. The last test that was conducted is the Western blotting technique, which was used to buffer cells and tissues. This is a very complicated procedure that involves buffering, boiling, blocking, washing, and incubating in mouse antibodies. Levels of the beta amyloid were then measured before and after the analysis. Using the methods shown on the previous slides, the result was that caveolin-1 is depleted in the brains of T2DM patients and in the brains of diabetic mice. On the first figure on the left, we observe a high level of CAV1 in normal, healthy, aging individuals, and a low amount of CAV1 in patients diagnosed with T2DM. You can also observe that there's a high amount of beta amyloid in patients with T2DM. The bar chart on the right also demonstrates the exact quantitative values of caveolin-1 in normal aging patients versus those diagnosed with T2DM. We also notice that the depletion of caveolin-1 in brains of diabetic mice promotes neuropathology and impairments in learning and memory. The first graph on the left shows a relationship between CAV1 proteins and amyloid precursor proteins. We can see as CAV1 decreased in DB-DB mice, the levels of APP increased in DB-DB mice. The wild-type mice had little to no APP, as shown on the figure. As we mentioned earlier, amyloid precursor proteins, APP, gives rise to amyloid beta peptides. Therefore, an increase of APP would correspond with an increase of amyloid beta, which we do see in this bar graph. We can go one step further and conclude that caveolin-1 does play a part in regulating the processing of APP. In this phase of the study, the novel object recognition test was used to test and impair hippocampal function in diabetic mice. In mice, we use a novel object recognition tasks to test amnesia, which is a partial or total memory loss and is a precursor for Alzheimer's disease. What the researchers found is that there is a significant difference in the scores of the novel object recognition task between the control mice and the mice that were diabetic, as seen in this figure. We can see that the control mice with a new object, here, have a much longer observation time than the diabetic mice with a new object, as shown here. In fact, the diabetic mice actually spent more time exploring the familiar object, here, than the novel object. Can the restoration of caveolin-1 in DB mice repair memory and cognitive function? This was tested by injecting diabetic mice with an adenovirus containing caveolin-1. Adenoviruses are really important in all sorts of experiments, especially those involving mice, because of their ability to infect dividing and non-dividing cells with the virus in any kind of tissue. After the mice were injected with a virus containing caveolin-1, they were tested in the novel object recognition task, and those scores were measured. As you can see on the figure on the right, the diabetic mice that were injected with caveolin-1 did better than the mice that were injected with the control. On the figure on the far right, we can see that the performance index was higher for the mice that were injected with caveolin-1. There were a lot of other smaller tests conducted within this one to test tau phosphorylation and BACE1 levels, as well as the targeting of other proteins which underlie the formation of the amyloid precursor protein. The results from there show that the various parts of the hippocampus and brain are elevated by the increase of caveolin-1 levels. Additionally, to determine if the amyloid beta levels would increase with the onset of caveolin-1, human embryonic kidney cells that express APP were used. It was determined that the levels of amyloid precursor proteins and amyloid beta are increased significantly when caveolin-1 is depleted. This experiment provides an important clarification and experimental model for discussion. This study proves that the reduction of caveolin-1 in type 2 diabetes patients is really important in the formation of the amyloid peptide protein, which is a big characteristic of Alzheimer's disease. Secondly, this experiment paves the road for scientists to explore the relationship between caveolin-1 levels and APP and amyloid beta levels. Additionally, this experiment gives rise to evidence that correlates the restoration of caveolin-1 actually helps memory and improve memory and cognitive function in diabetic mice. Last but not least, this experiment helps provide evidence that the reduction in caveolin-1 gives rise to the increase of cleavaging and secretases of APP, which you can see in this image, then give rise to senile plaques, which are an important component of Alzheimer's disease. This study is important for researchers studying the relationship between caveolin-1 levels and APP levels. It also provides a critical analysis of a scientific link between T2DM and Alzheimer's disease. There are a few limitations to this experiment as shown on the slide. Diabetic mice brains are not the same as T2DM human brains. Additionally, mice were very young in this experiment, and this could have an impact on the formation of amyloid precursor proteins.